Hello, I am Sama Goliaei and I am presenting our work Bayesian Phylogeographic Analysis reveals the effect of non-pharmaceutical interventions on SARS-CoV-2 lineage importations and dissemination during the third wave of the pandemic in Germany. This is a joint work with Dr. Furqman Arabi and Professor McCarty from Helmholtz Center for Infection Research. The goal of our study is to analyze the importation of SARS-CoV-2 lineages into Germany. The third wave of SARS-CoV-2 was a special case for this study. In this time period, systematic genomic surveillance in Germany has been started. Several non-pharmaceutical interventions applied in different states of Germany and also nationwide. Also, this period was before widespread vaccination in Germany. The goal of our study is to first infer the importations of the virus into Germany. As you can see in the figure, we tried to find the phylogenetic tree representing infection history of the virus. Then we found locations of the phylogeny nodes. In this figure, nodes with location inferred to be inside Germany are colored pink, and the nodes outside Germany are colored blue. A pink subtree represents a lineage imported and spread inside Germany. Root of a pink subtree represents TMRCA, and the icon of the person represents the importation event. Considering the number of importations to the country, we analyzed the number of importations in each day and evaluated the effect of MPIs on the number of importations. Here, I present the method which we use to infer the importations and the effectiveness of MPIs. First, we downloaded the genomic sequences of SARS-CoV-2 in June 2021. The dataset contained about 2 million samples. Then we subsampled the sequences with the number of samples proportional to the number of confirmed cases for each country in each week. Then we inferred maximum likelihood trees and refined the trees with a Bayesian approach with BEAST software. Then we applied another Bayesian discrete trait analysis, and based on its results, we inferred the imported lineages and their importation time. Finally, we defined an effectiveness measure for each day. We summarized MPIs in 12 major MPIs and evaluated effectiveness of them according to their application date and based on derivative of the number of importations to the country. In this slide, I present two results on the number of importations. First, we see the map of Germany, and states are colored according to the percentage of lineages first observed in them. As we see here, the states with high populations, North Rhine-Westphalia, Bavaria, and Baden-Württemberg, have the highest number of lineages first observed in them. We can see in this chart the number of imported lineages on each week. X-axis represents the weeks and Y-axis represents the number of imported lineages in each week. The colors show the state in which the lineages are first observed. As it is marked by red arrows, two peaks are observable after two holidays, Christmas and Easter. Here we can see the effectiveness of NPIs. The NPI with the highest effectiveness is the availability of free antigen rapid tests in March 2021. The second most effective NPI is obligation of FFP2 and surgical masks in public transportation and shopping areas. Here we see the conclusion of this short presentation. We inferred the events of importation of SARS-CoV-2 into Germany in the third wave of pandemic and evaluated the effectiveness of MPIs. First, We observed that most lineages are first appeared in the states with highest population, North Rhine-Westphalia, Bavaria, and Baden-Württemberg. This observation suggests to pay special attention to these states in future pandemic managements. We observed that the most effective MPI was the availability of free rapid antigen tests. Considering its low socio-economic impact, this could be a very good suggestion to be included for future pandemic management strategies. We also observed that two high peaks of the number of importations of SARS-CoV-2 into the country happened just after two major holidays, Christmas and Easter holiday. This also suggests special attention and application of special MPIs for the holiday times. Finally, I thank the two other contributors of this work, Dr. Foroghmand Arabi and Professor McCarty. Also, I would like to thank Professor McCarty's lab members. Thank you for your attention, and I would be happy to receive and answer any questions regarding this work.